Greetings for the dear students. Today I am going to start a new topic, that is metal ions in biology, and in this first lecture I'll cover the introductory part of the topic. So let's start metal ions in biology. A complex life. Life is a metal ion. Metals from S and G block acts as active sites in the majority of enzymes. Iron, zinc and copper are the examples. Metal containing biomolecules, they are very much important. Now we'll discuss the classification of elements. Now the elements can be classified as essential plus non-essential. That gives rise to all the elements. The essential elements are required for the maintenance of life of plants and animals. The absence causes death or severe malfunctioning. The non-essential elements, they don't play any positive role in biological system. Now we take the essential elements. Now they are bulk elements and trace elements. The bulk elements, they are the elements which are found in higher concentration, and trace elements, they are those elements which are needed in very low concentration. Now we come to toxic elements. The toxic elements are being arsenic, the average quantity being 18 milligram, and the excess quantity is cancerous. Similarly, Sn, the quantity is 6 and it is also cancerous. Bismuth, quantity is 0.2 mg, it is also cancerous. Te, the quantity being 0.1, this is also cancerous. Lead, the quantity being 121 and it causes plumbage. Cadmium, the quantity being 5.0, it causes high BP. Sb, the quantity being 8.0 and it causes poisoning. Be, the quantity being 0.04 and it causes heart disease. Chromium, the quantity being 1.5, it is also cancerous. The periodic distribution of biologically important elements is shown in this slide. Now we come to the metal ions in biology. They occur in several forms. The most advanced class is metalloenzymes or biological catalysts. They have a specific role. Major D block elements are included in it, but S block elements are also important, because the structural role, that is calcium, enzymatic action, magnesium, and homeostatic balance, that are sodium and potassium. Now we have the question, what are their roles? Let us see. The S block metals, they are bulk metals, sodium, potassium, magnesium and calcium. They maintain electrical charges, osmotic pressure, and they prevent the cell from collapsing by making it turgid. Now we come to the sodium ions. Major cation of the extracellular fluid. It creates electrical potential for the functioning of nerve cells and muscle cells, and the concentration being 0.01 molar. Sodium ions also serve as essential activating ions for specific enzyme activity. They are associated with acid-base balance. Now we come to potassium. Potassium's concentration being 0.15 molar. It maintains electrolytic balance, proper functioning of heart, maintenance of blood pressure, nerve impulses, aids to the waste removal process, enhances the muscle functioning in synthesis of ribosomes. Now we take the transportation of ions. Charged ions must pass through the hydrophobic membrane. Neutral gases, that is oxygen and carbon dioxide, and low charge density ions, that are anions, can move directly through the membrane. The high charge density cations require help. Now what is the mechanism? First we will discuss ionophores. They are special carrier molecules that wrap around metal ions so they can pass through the membrane by diffusion. Second is ion channels. Large membrane spanning molecules that form the hydrophilic path for diffusion. Ion pumps, molecules using energy to transport ions in one direction through a membrane. Now we come to the types. There are two types. Number one is passive transport. It moves ions down the concentration gradient requiring no energy source. Ionophores and ion channels are passive. Active transport. It moves ions against the concentration gradient requiring energy from ATP hydrolysis, that is ion pump, are active. Now the choice of transport mechanism are the charge, size and ligand preferences. Now we will discuss the sodium and potassium ion pump. Sodium potassium ATPase, membrane spanning protein ion pump, alpha 2 beta 2 tetrameric 294,000 Dalton protein. Conformational changes pump the ions. One conformation binds sodium best, the other binds potassium best. Hydrolysis of ATP provides energy for conformational changes. 30% of a mammal's ATP is used in this reaction. Now, antipode transport. Like charged ions are transported in opposite directions. Reversing the normal action can generate ATP. Reaction can occur 100 times per second and the reaction is shown in this equation. Sodium and potassium ionophore. Non-action microbial sodium and potassium ionophore makes sodium ions and potassium ion membrane soluble when complexed. Oxygen donors can be modeled by crown ethers. Here I have shown the structure of the crown ether. Sodium and potassium ion channel. Dramamine, the ion channel forming molecule. Helical peptide dimer, hydrophobic outer surface interacts with the membrane. Carbonyls and nitrogens are on inner surface can interact with cations as they pass through. Potassium selective, that is pore size and ligand select for potassium ions. Channels can be voltage graded or activated by binding of a chemical effector which changes the conformation. 10 raised to the power to 10 raised to the power 8 ion per second may pass. Here I have shown the potassium ion channel and the graded sodium channel in this diagram. Now magnesium ions. They are found in chlorophyll, intracellular fluid. Carbohydrate metabolism enhances nerve impulses. Extracellular fluid, active site for transferase, phosphohydrase. Calcium ions. Their function being bone formation, brain functioning, maintenance of heart rhythm, blood clotting, formation of teeth enamel, that is fluoroapatite, 1,000 times less in intracellular fluid. Here I have shown the calcium rich foods and magnesium foods. Now we come to the D block elements. First is iron, next is vanadium, chromium. Vanadium is extremely small amounts it is needed. Lichens and fungi have it in the active site of enzymes. Appears in nitrogenase vanadium in the haloperoxidases. Chromium. Chromium third is found in mammals, lipids and glucose metabolism, glucose tolerance. Chromium 6 and chromium 5th, they are mutagenic and carcinogenic. Chromium 5th damages DNA, normal selection of insulin. Manganese. In wider range of organisms, it is found in important enzyme processes, photosynthesis. Amine 2nd and amine 3rd have redox role. For example, the enzyme superoxide dimutase, SOD. Iron. It is truly ubiquitous, required for tissue growth, responsible for oxygen transport. In humans, hemoglobin and myoglobin represent 65 to 6% of all iron in body. Ferritin is rapid access of iron. Something about ferritin. The structure. Symmetric spherical protein coat of 24 subunits. Subunits are 175 amino acids, 18,500 Daltons each. Channels in threefold axis of hydrophilic iron entry inside the surface of all hydrophilic. Inner cavity. 75 angstrom inner diameter holds 4500 iron atoms. Iron is stored as ferrihydrate phosphate. Iron protein interface. Binding of core to protein is believed to be through oxy or hydroxy bridges. Here I have shown the structures of ferritin. Now in this slide I have shown the pathway of Fe2 plus from blood stuffs to hemoglobin, and ferritin involves the mechanism. Following mechanism, in this slide we can see the mechanism. Now cobalt. It is essential in small amounts. Cobalamines are found in many organisms, 5 mg in humans. Enzymes with inert cobalt third has been discovered. It provides labile reaction sites. It is extremely toxic to plants. Next is nickel, an important component of urease, carbon monoxide dehydrogenase and methyl S coenzyme M reductase. It exists in oxidation states like first, second and third. Copper. It is present at active site of a large number of enzymes. Third most abundant transition element, as it acts as electron transfer agent. For example, cytochrome C oxidase, tyrosinase, nitrite reductase. It is blue color protein type 1 in hemoglobin formation. Zinc. The quantity in adults being 2 to 3 grams. Carbonic anhydrase, carboxy peptidase, 300 known enzymes, DNA binding proteins. Zinc reserves, maintains normal concentration of vitamin A, tissue repairing and wound healing. We can get it from these sources, that is the sources of zinc. Now in this slide I have shown the symptoms of elemental deficiency in humans. Calcium deficiency results in retarded skeletal growth. Magnesium deficiency, muscle cramps. Iron deficiency, anemia, immune disorders. zinc deficiency, stunted growth, skin damage, and retarded maturation, copper deficiency, liver disorders and secondary anemia, manganese Mn deficiency, infertility, impaired skeletal growth, molybdenum deficiency, retarded cellular growth, cobalt deficiency, pernicious anemia, nickel deficiency, depressed growth, dermatitis, chromium deficiency, diabetes symptoms, silicon deficiency, skeletal growth disorders, fluorine deficiency, dental disorders, iodine deficiency, thyroid disorders, selenium deficiency, cardiac muscular weakness, and arsenic deficiency, impaired growth in animals. Ultra trace elements. They are needed in less than 1 ppm or 50 ppb. It includes arsenic, boron, silicon, nickel, vanadium in humans. Other elements include Br, Cd, F, Pb, Li, and Sn. Quite toxic at any concentration much above the concentration ultra trace level. So why carbon instead of silicon? The question is, why carbon instead of silicon? The conclusion is life revolved utilizing these elements that are abundant and available to it and become dependent upon them. Rare elements are not utilized. So this was all about this topic. Thank you. Have a nice day.